So I would like to start with introducing our first candidate here. We have 3 presentations today. And the first one is Ahmad Alhomodi from South Dakota University here in the USA. The title of his talk is Applications of Fungal Mycelium, Co-Cultures of Fungal Mycelium during Solid State Fermentation of Canola Meal for Potential Feed Application. I'm very excited to hear this talk because canola meal is a vastly underutilized substance and there's a lot of it. Ahmad is currently pursuing a PhD and he is focused strongly on canola meal extraction. Ahmad, would you like to start?
Good morning, everyone. First, I would like to thank all of you for attending my presentation, which is under the title Application for Co-Culture of Fungal Mycelium during Solid State Fermentation of Canola Meal for Potential Feed Application. Canola is an important oilseed crop, which is ranked second after soybean. When canola seed is a process for oil extraction, 2 fractions are generated. We have oil fraction and meal fraction. And it is estimated that for each 1 kilogram of oil produced, 2 kilograms of meal generated. And oil has a different industrial applications such as human food and biodiesel, while meal is used in animal diet. If you look at the canola meal benefits, even though the meal is rich in protein with well-balanced amino acids, its inclusion in animal diet is limited due to the presence of anti-nutritional factors such as glucosinolate, phytic acid, high fiber, and oligosaccharide, beside low protein digestibility. So the bioprocessing goal is to enhance the nutritional and the commercial value of canola meal by increasing the desirable components such as protein and decreasing the undesirable components such as anti-nutritional factors. If you look at the possible solution, several attempts have been done to improve the canola meal, such as milk pretreatment, microbial fermentation, pretreatment followed by microbial fermentation. All these processes showed the potential in decreasing the anti-nutritional factor, but fibers is still a challenge. The drawback of using tail end dehulling process is the high energy input requirement beside the loss of protein in the whole fraction. Thus, our project is to exploit the synergistic relationship among 3 different fungal strains, Aerobacillus bolonensis, Neospora crassa, and Trichoderma russii, in co-culture incubation under solid state fermentation to improve the canola meal composition. In our material methodology, we started with a canola meal of 50% of canola meal on dry basis. The meal was autoclaved and then inoculated with the mono and co-culture of 3 different fungal strains, Aerobacillus bolonensis, Neospora crassa, and Trichoderma russii, beside an inoculated control. All the flasks were statistically incubated at 30 Celsius for 168 hours, and at the end of fermentation, all the samples were dried, grind, and analyzed for anti-nutritional factor and crude protein. Here in our obtained results, before I start talking about my result, let me tell you what the abbreviation are. If you look at the first row, we have TSR, which is a total solid recovery, CP, crude protein, TC, total carbohydrate, CF, crude fiber, ADF, acid digestion fiber, NDF, neutral digestion fiber, GLC, glucosinolate, PA, phytic acid. If you looked at the first column on the left, we have HECM, which is hexane extracted canola meal. And at the bottom, we have AP, which is apollonans, NC, Encrasa, TR, Trichoderma russii. So in our results for the monoculture, Encrasa showed the least total solid recovery, and it was the best in increasing the protein content and reducing the crude fiber, acid digestion fiber, and neutral digestion fiber. Whereas the apollonans was the best in reducing the total carbohydrate and the glucosinolate, but russii was the best in reducing the phytic acid compared to the other fungi. In the co-culture fermentation, the combination of apollonans And GRAS showed the least total solid recovery, and they were the best in increasing the protein, reducing the total carbohydrate, reducing the crude fiber, reducing the acid-digen fiber and neutral-digen fiber, whereas ebullonants with TRCI were able to reduce the glucosinolate and phytic acid. So in summary, all that strain individually and co-culture increased the protein compared to the raw meal. The combination of ebullonants and GRAS were the most effective in reducing the fiber content, and the co-culture of ebullonants with TRCI were the most effective in reducing the glucosinolate. So in conclusion, the co-culture and fermentation improved the canola meal composition, but that improvement is different based on the fungal combination. So the selection of microbe or the fungi need to be based on the goal of the final product. And finally, this improvement in canola meal could help in solubilize the protein and increase the protein extractability, which is considered a big problem in the protein extractability that we are currently studying. That leads me to the end of my presentation. Thank you for your attention, and I will be pleased to take your question.
Thank you, Ahmad. That was very interesting. So now we have a question in the chat, and we have our judges here. Judges, do you have a question to start with?
Yes.
Professor Ferrer-Diaz?
Thank you, Ahmad. It was a very nice presentation. Congratulations for your work. Very interesting. But I have a question because you tried the strains isolated and 2 by 2, and also all the 3 together. But in almost, I think, for all the parameters you followed, when you use the 3 strains together, the results were worse than using only biculture. Can you have an explanation for this, or not yet?
Yeah, that is a very good question. It's based on because when I use two microbes together, sometimes the product of one or these 2 microbes could inhibit the other microbe, for example. So for the TRCI is known for the production of cellulotic enzymes, but the presence of many like a sugar and the soluble sugar in the media cause a feedback inhibition. That's why you see that there's no good result when they are together, because if there's a lot of sugar, for example, it's because of feedback inhibition for this microbe. There was thus the fermentation is not like when there's just 2 microorganisms.
Okay, thank you. Thank you.
I'd like to ask a question. So I'm Doug Hayes. Thank you, Ahmad, first of all, for giving the presentation, and very good work, very interesting. Just a short question. Have you looked at any process economics, or is that something that you're going to be looking at later on in your dissertation work?
Yep, that's a great question. We haven't looked at that yet, but I mean, what we are using is not that, costly. And I think in the future, we have to look at that when we want to scale up and see how much that could be cost and what is the process cost. Yeah, but currently we haven't done that yet.
Yeah, actually it's a more difficult question than anybody ever tells you about when you're in graduate school. You have to get engineers involved, and generally one of the interesting things to think about is you really want to adapt some other process that's already been costed out, so people know what it's going to cost, and that would be very. I'm a very big believer in canola meal utilization, so this is exciting. We have an opportunity for new, enriched enriched products this way. One question I was wondering about was, how did you select your co-culture strains? Obviously, there are millions to choose from.
Yeah, all the fungi that we have are generally recognized as fungi. And we have a mini collection, and we choose these microorganisms based on their enzymatic ability, based on the previous studies, to see if and also we did apply a dole culture to test any antagonistic behavior among these microorganisms before we use them. And we found that they grow together without any antagonistic behavior. And we saw that their ability in producing a cocktail of enzymes, that's why they were selected based on that. Yeah.
Excellent, thank you. Well, I don't see any other questions unless any of the other judges have a question. Thank you very much, Ahmad, for a very interesting talk, and I wish you great success in your future.
Thank you very much.
I'm sure you'll tangle with many interesting problems.
Thank you very much.
Okay.
Thank you.